In this video, I'm going to discuss some key concerns when working in the biopharm field as an operational or quality professional. I will highlight the key differences between biotech products and APIs that are manufactured using chemical syntheses. From this information, I want to inspire you to take a fresh look at this field of pharmaceutical manufacturing and put these learnings into action in your workplace. Here are the 5 key differences with biotech products. Most often, long-chain protein structures are fragile and special measures are needed to produce and protect them. Long-chain protein structures can be immensely difficult to characterize. As a consequence, product quality assurance is hugely dependent on effective process control within scientifically derived process parameters. Management of change is critical. Remember the FDA's historic statement of intent: the product is the process. Biotech production is all about cultivation of the required protein under monosepsis and then systematic separation of the impurities, quite different to a typical chemical synthesis. So let's look at each of the 5 items in turn. If your typical chemically synthesized API is considered to be robust, resistant to external change, and easily understood, think Arnold Schwarzenegger. Yet your typical long-chain protein structure is complex, it's hard to characterize, and it's prone to unexpected change and is a bit weedy. So think of David Beckham or Lady Gaga. Therapeutic proteins like divas need and demand special treatment. Our quality management systems need a different approach if we're able to make high-quality, stable biopharma products. Secondly, characterization of long-chain molecules is very, very difficult. Small-chain molecules can be assayed by devising QC methods that seek specific chemical bonds or specific single-activity sites. For instance, looking at the structure of a paracetamol molecule, how easy is it to test for bonds, structure, and groups on this molecule compared to those on protein structures? Look at the vitamin B12 structure and a typical monoclonal antibody. So if you are limited in detecting impurities, structural errors, and other degradation products, what can you do to control product purity? The answer is to build quality into your product by effective design, control, and monitoring of the production process. Quality by design is critical in biotechnology and is the only way to effectively detect and minimize product variation. Control your process and you will control your product quality. This is why stringent process design, comprehensive process validation, and flawless execution of each step in the production process are critical to the quality and stability of biotech products. So you heard it before, the product is the process. But what does this really mean? Put simply, if the manufacturing process is subject to variation, a variation in the product is to be expected. Product variation reduces the assurance of product quality, and any variation has to be detected, assessed, and minimized using whatever technical and procedural strategies are available. Making biotech products with minimal process variation is key to minimizing important side reactions that cause protein impurities that then reduce product yield or alter the safety profile, bioequivalence, or bioavailability of the drug. In short, if you aren't in control of your process, you're not in control of product quality. So finally, what might a bioprocess look like? Take, for example, a biotech product produced during highly controlled fermentation of a host cell such as E. coli. Within the host cell, millions of different proteins are generated during fermentation, with one particular chain design and structure having the desired therapeutic activity. All of the others, either inactive impurities or potentially toxic impurities, are unwanted. Strict control of fermentation maximizes the generation of the desired protein to the detriment of the undesired protein. Downstream processing then seeks to break open the host cell, often by centrifugation, and separate the desired protein from everything else via, for example, various forms of chromatography and high-tech filtration. Bioprocessing can be a long, complex and multi-stage chain of activities, all subject to a multitude of in-process checks, tests and monitors. To those of you more used to chemical syntheses, it can look formidable, both in terms of performing GMP audits, performing investigations into non-conformances, and approving batch records, validation protocols and technical reports. Our expertise can help you to make the right strategic decisions, make the right tactical calls, document the approach taken, and embed the learnings. Our expertise is on hand to help you improve your company performance, meet GMP expectations from clients and regulators, and ensure product attributes are in place, always keeping the end user in mind. We can be summed up as the right people, the right solution, the first time.